Okay, so if we want to find out how this reactive oxygen species involved in a process of immune response or inflammation, it's a one thing is a beneficial effect. So we have learned in the previous paragraph the reactive oxygen species is an inevitable byproduct. The formation is inevitable, so we can't avoid this one. It's anyway happening. It's a natural process. Free radicals are formed. The bad thing is if it is formed too much, this leads to oxidative stress. It's too much produced. Now, why it is called oxidative stress? Because now when we consider the stress is something we all experience at certain level. We can't avoid this. So we use this term stress, that it is very uncomfortable conditions arise if we have to face certain challenges. When you have exam, you get stress. And what it says that stress is a driven factor uh, which we force to do it well. But too much stress is harmful. Same way, the oxidative stress is harmful for the cells if it is produced too much. Otherwise, the free radical formation is beneficial for immune response and inflammation. And the other two things are synaptic plasticity and learning and memory. If we are asked at least how it involved in immune response and inflammation, uh, this is the answer. Now, in both the immune response and inflammation, there is a process we learn as the phagocytosis. So, if this is a phagocytic cells, the phagocytic cells can engulf pathogens, and after engulf the pathogen, it forms this structure called the phagosome, and the phagosome is fused with the lysosome, right? So this is a lysosome, and this is a phagosome. After that, lysosome secrete enzymes. These enzymes can kill the pathogen. So we have learned lysosome release enzyme. Then the enzyme can kill the pathogen. Uh, they can also use reactive oxygen species to damage the pathogens. Okay, at the end, kill the pathogen. Reactive species can use to kill the pathogen. The short answer is reactive oxygen species can be used to kill the pathogen. So immune response and inflammation, reactive oxygen are useful, so they can even inflammation when they want to clear the damaged molecules and particles, reactive oxygen species can be used. And also to kill the pathogen, reactive oxygen species can be used, right?
So
So,
even what about the T killer cell? T killer cell can't it release these ROS?
well, we don't learn actually in the theory section what is the role of reactive oxygen species. But in case for the safe side, if you want to know what is the exact role of reactive oxygen species, we search ROS in immune response.
So I can't see the screen.
Okay, I just googled. ROS have been implicated in many aspects of immune response to pathogen, mainly related to innate immunity. Indeed, they have been proposed to be the common determinant of inflammation activation, which is critical in the inflammation process and the necessary for efficient immune response. Rows are also essential. You see here, rows are also essential for pathogen killing by phagocytic cells. I think this is well-known function of rows. I don't think that there will be a questions too much about rows and inflammation or immune system because we don't learn in the syllabus the rows and the inflammation or immune system, but we can better prepare for the scientific article if they say rows are important on immune response. Based on this sentence, we should learn what is the function of rows. Generally, rows can kill the pathogen, but T cells and B cells, it's a different mechanism. So further, if you look this a diagram, the mitochondria is here, and the mitochondria produce reactive oxygen species, and they may be important for many other functions. But our level, it is enough to know that the reactive oxygen species can kill the pathogens. However, when produced in excess, those molecules can induce oxidative stress, damaging proteins and DNA and inducing lipid peroxidation with the corresponding mitochondrial structures as the first target of toxicity. Okay, now other function of reactive oxygen species produced in excess, they can directly damage the molecules like protein, DNA, and proteins, lipid. So if they damage proteins, enzymes are prime target, transporters are prime target, so they can't work properly. If a DNA damage happen, this may induce mutation or prevent gene expression, switch on and switch off. And the lipid peroxidation is very bad because membranes are affected. However, the first target site is mitochondria because reactive oxygen are formed inside the mitochondria. There the concentration is high. So it has more reactive oxygen species. Mitochondria are vulnerable for first target of damage by reactive oxygen species. Furthermore, since mitochondrial DNA is localized close to free radical production site, I think in the past exam paper there was a question: what is the difference between mitochondrial DNA and nuclear DNA?
The
So
mitochondrial DNA is like a circular DNA, right?
yeah, uh,
Uh,
circular
it
and
doesn't have helical structure. Does it have introns and exons? It doesn't, right?
well, I think another important difference
Or
is
is
they
it
are not protected with
nuclear?
histone. They
Right.
don't have histone
Mm.
because histone, one hand protective proteins. The nuclear DNA has histone. Mitochondrial DNA has no histone. Mitochondrial DNA is circular, and mitochondrial DNA, nuclear DNA is linear. At least two differences we can clearly highlight. If they ask a question, write two difference between mitochondrial DNA and nuclear DNA, these 2 points definitely get 2 marks. And this mitochondrial DNA is localized close to free radical production site. Now, where is the free radical production site? There in the electron transport chain. What is the function of mitochondrial DNA? Mitochondrial DNA produced enzymes and the proteins which are mostly important in the electron transport chain. So, one hand, we can also understand that they are very close to the place where it produces these proteins because mitochondrial DNA give codes to produce enzymes and transporters of electron transport chain. They are proteins. So they are very close to each other. So free radical damage can have effect on this circular DNA, and they are not protected with the histone. So they are more target for the free radicals because they have no protection. So oxidation stress can trigger cell death and has been implicated in the pathogenesis of many neurodegenerative diseases such as Alzheimer's disease. So Parkinson disease we learn in the syllabus. Parkinson disease is reduced dopamine secretion by certain neurons, and these neurons located in the place called substantia nigra. The cells are dying. When the neurons are dying, they cannot produce enough dopamine. As a result, dopamine cannot, the lack of dopamine can cause synaptic transmission, impaired synaptic transmission, and people get mostly the weakness of the muscles, and they cannot coordinate the muscles, the motor function lost, so they develop mostly the uncoordinated movement of the hands and legs and other part of the body. But Alzheimer disease, we don't learn specifically. Alzheimer disease is again loss of neurons. This has direct effect on the memory. So they have memory loss, and this come under the condition called dementia. So dementia is condition the loss of memory progressively, and Alzheimer's seems to be one of the important disease for dementia. Just even though we don't learn much about Alzheimer, a growing body of evidence highlight bioenergetic impairments as well as disturbance in the reduction oxidation homeostasis in the brain with increasing age. Now this bioenergetic mean ATP production, and this is driven by this oxidative transport electron transport chain, which is series of reduction and oxidation steps, right? So the broad sense the homeostasis is maintenance of constant internal environment. So it should maintain a right amount of energy production. At the end, if there is a damage, we can measure this damage by measuring the outcome of energy production. So it lost energy production can decrease if there's a loss. But this is again inevitable because everybody die, because aging is unstoppable. Everybody get aging. With the aging, the molecular damage take place because it's a long-term accumulation of reactive oxygen species can damage the cells of the brain. So it's a natural process, but this natural
So
process.
the the energy given out by the mitochondria can be measured to find out the damage caused to the mitochondria, or like the energy given out by the cells can be measured to find the
Well, yes, one hand, yes, because if you remember when the brain imagings like CT scanning, uh, nuclear magnetic resonance imaging, uh, and then PET scanning, positron emission tomography. Now, if we look these three type of imaging method, the computerized tomography and magnetic resonance imaging and also fMRI, that is functional magnetic resonance images. The theory of these two based on the consumption of energy. So, for example, positron emission tomography, we inject radio labeled glucose molecule, and the localization of glucose molecule by the cell scanned by the scanner, and radioactivity is detected. So what is the contrast? So radioactivity is high, that means cell consume more oxygen. So if a brain area is like this and a healthy brain is like this, so if we detect healthy brain, this area, the radioactivity is high. Why the radioactivity is high? Cells are normal, they take oxygen, the oxygen is used. But if we take a diseased area of the brain, the radioactivity is low. It's like a black now. So this is a white, this is a black. Why is that? The cells use less amount of glucose. If less glucose is used, less oxygen is required for the aerobic respiration. So we can detect this kind of a thing, the brain imaging. I think there is a very good chance of asking, suggest how do you measure if the brain is used if the brain is produced less energy, how do you detect this? So positron emission tomography is a one thing, and then fMRI can also use, because fMRI, which couple this MRI couple the radio waves and the magnetic field. So this is radio wave and magnetic field. And functional MRI, it's added also oxygen consumption by hemoglobin. So that again very important. The functional MRI used oxygen consumption by the cells by hemoglobin. So both fMRI and PET are the best method to
So like
detect.
there's a close relationship between the fMRI and the the thing they are talking about in the passage. Like fMRI uses oxygen, and RO is can also that relationship is also there. Like they might ask more of fMRI. I feel like.
Yes, even most of the diseases, Alzheimer, Parkinson, and other diseases, they have to use fMRI and positron emission tomography to check the activity of the neurons. So cell death is tracked by less activity of the cell. They are black color. So when they get black color compared to the healthy brain, the cells are inactive because they produce less. They don't use. They are not producing energy. If they don't produce energy, oxygen is not used, and this can be tracked by glucose or oxygen level using fMRI and PET. So I think if there's a question, definitely. I mean, I hope there will be question about the scanning. Then check which one you're going to use, either PET or fMRI. However, if we ask another question, why energy requirement is high? Excitable cells. Now, here the brain is remarkable organ composed by highly differentiated cells that populate different anatomical region. Okay, in case the differentiated cells. Now, this can be again a little bit tricky. That kind of a question. We have to use stem cells, cell differentiation, genes on and off, like all the stories we have to bring here, because neurons cannot divide the after differentiated, the cell cycle stop because the genes are switched off. The genes are switched off. We cannot switch on the gene to again produce. And one therapeutic approach we learn at the stem cell level, learning theory, stem cell can be used to treat neurodegenerative diseases, because stem cell can differentiate into neuronal cells and they can replace with the dead cell. So they can replace the dead cell and they can function again. But the basic concept of differentiation, I think, in case we learn, differentiation is due to the surrounding chemical stimulation. This lead switch on, switch off the genes and epigenetic modification. As a result, they produce a protein by transcription and translation. This protein can permanently change the cell. This is again AS level theory, how cells are differentiated. And differentiated cells have specific functions and they located different part of the brain. So when they say different anatomical region, that means our basic knowledge is cerebrum, cerebellum, and medulla oblongata. The other three parts we learn. and they have a different functions because cerebellum is important for the coordination movement, and the cerebrum is important for the higher functions, learning, memory, speaking, hearing, visions. And the medulla oblongata, mostly we have learned the regulation of involuntary function like heart rate and the respiratory rate, like things are important. So these functions are, they can do these functions because cells are differentiated, and they can't change it again. Neurons are polarized cells with different morphology. So polarized mean we learn that they can maintain the some kind of positive and negative charge outside and inside the cells. So resting membrane potentials, outside is positive, inside is negative. So this polarization is made by different distribution of ions. According to their morphologies, we learn some of the neurons like motor neurons, relay neuron, and sensory neurons. According to their role and their localization, these pose mitotic and excitable cells. So excitable mean they can change into depolarized stage, resting membrane, move to the depolarization stage, and then repolarization. So they can do this kind of changes, really high energy requirements. Now, why they need high energy requirement? So you can recall what we have learned. This is the sodium channel. Sodium moves in, and this is a potassium channel. Potassium moves out. And the third one, sodium potassium pump, again takes sodium out and potassium in. For that, it is required ATP. It's active transport. Now, imagine this is happening all the time. This is happening person born to the death. And how many neurons in the body? Like billions of neurons. So billions of neurons, and maybe this kind of a trillions of transporters. Every single second, every single time they are using this ATP, the energy requirement is high. But other cells, they use energy when there's a requirement. For example, muscles are excitable cells, but we learn ATP is need when muscles are contracting. So they use a lot of ATP when there is a contraction of muscles. If we take normal cells like endothelial cells use ATP for specific functions like protein synthesis, maybe active transport when there's a requirement. But neurons, in addition to their housekeeping functions, like regular functions, they have this additional role. Continuously, they maintain this polarization. For that, they need ATP. So that's an important point. In case there is a question why energy requirement is high in these excitable cells. So you can mention this sodium potassium ATPase pump is continuously working to maintain resting membrane potential. In addition to that, excitations and the depolarization and repolarization to open up some transporters, they can also use ATP, and also exocytosis need ATP. However, uh, they have actually given in the article why they use ATP. Because I thought that when we have this paragraph, definitely there's a question explain what are the function of ATP in these excitable cells, like production, maintenance, secretions, neurotransmissions. And here it's given as a four function to maintain their membrane potential, allowing the propagation of electrical signals. Now, further, if you ask how they maintain membrane potentials, Again, you have to say sodium ion moves in, potassium ion goes out, and sodium potassium pump maintain the outside positive, inside negative that need ATP. Then the re-establish of ion balance after the firing of action potentials. Right, and funny thing is, again they given this sodium potassium ATPase activity. So answer is given. I mean, I don't know whether there will be a question because answer is given here. Third one, to trigger the release of neurotransmitter by exocytosis. It is also given. So I mean, it's a good question, but answer is given. Membrane polarization, and then the exocytosis, and fourth one, allow the recapture of neurotransmitters from the synaptic cleft. Now, if you ask, I mean, they have given, but if in case there is a possible question, if you ask one any of the event, like now, explain how energy is required for the recapture process. Now, how energy is used for recapture process? It's again, if these are the neurotransmitters in the synaptic cleft, so what happened? Membrane is using this endocytosis method. Sorry, endocytosis to recapture the neurotransmitters. For endocytosis, it again need energy. It is an active process, right? Endocytosis. So this one, endocytosis, to trigger the release of neurotransmitter by fusion of this is we learn again exocytosis. This is endocytosis, and this one, second one, and first one, it's again given sodium potassium ATPase activity. So since sodium potassium ATPase activity is given here, there may be a chance asking third one and fourth one how energy is used for these two processes. It is exocytosis. Third one is exocytosis. Fourth one is endocytosis. You have to mention this word. You have to explain how energy is used for this kind of a thing. Okay, glucose oxidation is the most relevant source of energy in the brain. Since other fuel sources such as fatty acid oxidation have an ATP generation rate too slow to sustain neuronal energy demand and produce too much loss that may cause oxidative stress. In consequence, neurons rely almost exclusively on the mitochondrial oxidative phosphorylation system to fulfill their energy needs, uh, supplied under the form of ATP. Now, brain is use fatty acid. Uh, the generation rate is too slow. Generation rate is too slow because it need different pathways. So we have learned glycolysis, link reaction, Krebs cycle, and electron transport chain. Now this is a standard pathway of oxygen molecule, glucose molecule. So when glucose enter, it is going through this pathway. If a fatty acid enters, then the fatty acid has to break down into two carbon compound, and then again it enter at the level of Krebs cycle. So you can understand the fatty acid need additional pathways if they want to fulfill energy requirement of the cells. So ATP generation is rate is slow. And why it can produce too much rose? Because maybe one reason fatty acid has long hydrocarbon chain. It has more carbon and hydrogen bonds. So it has more carbon and hydrogen bonds than glucose. There may be a reason for that, having too much carbon and hydrogen bonds, but their energy level is high, also reactive oxygen is also high. So, a main fuel for brain or neuron is glucose and oxidative phosphorylation. I have taken this from the textbook. So, as I mentioned, fat, you can see a fat break down into fatty acid and glycerol. So this fatty acid, if it has 16 carbon, so then it can break down into 8 carbon, 8 times 2 carbon molecule, because acetyl-CoA has a 2 carbon, then they enter the Krebs cycle. Glycerol can enter through the glycolysis, and if a protein is energy source, amino acid, again into the level of pyruvate and acetyl-CoA. So glucose seems to be the ideal molecule because it gives always the standard pathway. But other source like fat and protein wants to provide energy. They have to enter into another pathway to produce smaller molecule like 2 carbon molecule. And again they can enter to the glucose oxidation pathway. Right, something to keep in mind how the other respiratory substrate involved in energy production. Now, some studies aim to compare mitochondrial properties in different organs, including the brain. For instance, mitochondria isolated from rat liver, kidney, brain, and skeletal muscle shows significant and similar uh, proton leak, but the phosphorylating system appeared to more active in the brain and the muscles. So in this rat experiment, they use different organs: liver, kidney, brain, and skeletal muscle. Because different organ energy requirement is different. So when the energy requirement is different, they can check if there are any different with rose production. The conclusion is similar proton leak. Now, what does it mean? Proton leak is when NADH and FADH reduce oxidize to its oxidized reduced form. The proton is released. Hydrogen ion is released. It's a proton, and then energy is used to pump proton into the intermembrane space. So this is called the proton leak. The proton leak is considered as pumping hydrogen ion out. But the phosphorylating systems appear to be more active in the brain and the muscles. At the end, we learn this hydrogen is moving back to the ATP synthase complex to make ADP plus phosphate into ATP. So this is the phosphorylating system. This is very active in brain and muscles because energy requirement is higher for brain and muscles. Both are excitable cells and they need a large amount of ATP. So if you are asked what is the proton leak, you should mention NADH and FADH oxidize